Thank you. It's an absolute pleasure to be here. And I think you can see from the videos, we had a lovely time. Was that 2019? Yes? Because nothing happened in 2020, no. So we always keep making that mistake. So it's a real pleasure to talk to you today about matching and genetic panels. So I'm Professor of Reproductive Science at the Institute for Women's Health at UCL, and I'm always glad to come to Cyprus. There's many of my alumni here, and it's always great to catch up with you. So just some disclosures. I run a distance learning certificate in clinical embryology for embryologists under the Embryology and PGD Academy. And I run a Facebook and website called Global Women Connected, where my passion in recent years has been to educate women and men about women's fertility and women's health issues. And I started my career as a clinical embryologist in 1987, and at that time, I realized that my friends didn't understand about their menstrual cycle or about their fertility. And I started writing a book on fertility, which I finished this year. So, thank you. I am writing another book. It can't take 30 years because I think I might be dead. So I'm going to do it hopefully do it a bit quicker. But it's called Your Fertile Years, and it's for the public to understand how women's fertile years work. So it's from puberty to the menopause. But I've been paid by these companies or done some work with these companies over the last few years. So what I'm going to talk to you about today is mainly some work that I've done with Cryos on genetic testing of our donors. So we published this paper recently, and I'm going to share that data with you today. But I am going to touch on the importance of ancestry testing, which is very relevant after our talk, well, several talks. It's come up in several talks. But I spoke more about that in 2019, so I'm not sure if those videos are still available, but you can hear more about that talk in more detail in the video. And I want to just say a few things about my view about talking to our children about their conception. And again, I think from Savio's talk this morning, that again came up, so it's really important. I'm going to briefly talk about the fertility education, and then I'm going to leave you with something really controversial, which follows on from Evelyn's talk about where the future of eggs and sperm is actually going. So I'm going to leave some real crazy ideas with you, which I actually cover in the last chapter of my book. It's about the future of reproduction. So let's talk about genetics first. So I've been really lucky that there's a group of us between ESHRA and the European Society of Human Genetics that have had 2 meetings, and we're experts in fertility and genetics. And in those 2 meetings, we brainstormed about the issues around genetics and fertility and what we thought we should be discussing. So our last paper was in 2017, and I've been trying to push ESHRA to have another meeting, but it really needs to be a face-to-face meeting where we can really discuss the ideas of what's happening in the fields of genetics and fertility. So I'm hoping that next year we will have another meeting, and then we will write another update. But some of the issues we've talked about today already, such as expanded carrier screening, are written up in this paper, and we've introduced those topics there. So every one of us in this room does carry some mutations. If we all got sequenced, has anyone been sequenced? I haven't been sequenced. I'm like Alan, I'm too scared. A hand's just gone up. Well done, well done. Very brave. I have done 23andMe, and I learned some things under 23andMe which I didn't know. A major thing about my ancestry, which was a revelation to my family who didn't quite believe it, but more have been tested and we have to believe it. But going to genetic disease, there are thousands of genetic diseases, but they're all quite rare. And as I said, all of us in this room carry mutations. If we all got sequenced, the latest data is suggesting that we carry maybe up to 400 mutations in our genes, but most of these are what we call recessive. So just having one abnormal copy of that gene is okay. We don't have the disease. But if our partner has that mutation as well, or a mutation in that gene as well, then we're at risk of transmitting that disease to our children. So these recessive diseases are now becoming really important for us to think about, should we or should we not do any genetic tests? So we could pass them on to our child. But if we're going to do any analysis for a recessive disease, we really need to look at the couple together, or the donor and the recipient together. So I just want to do a little bit of revision of genetics. I always say that anyone working in fertility really needs to understand genetics. So hopefully this is just a revision of what you did at school or more recently. So a recessive disease, if you see the circles are women in the family and the boxes are men. And if there's a dot in the middle, that's a carrier. So in this family here, the recessive family, the parents at the top were carriers. They had children who were carriers. But when those two children those two people had children together, since they're now both carriers, they were at risk of transmitting that disease to their children. So they had a 25% risk of a child being a carrier, a 25% risk of that child being affected, and then a 50% chance that the child could be affected with the disease. So, well, the first one is a carrier, the second one is unaffected, and the last two are affected with this disease. So you can see there how this spans out within the family. For a dominant disease, you can see here that whether it's male or female, you only need one abnormal copy of the gene. So it just goes down the family no matter what the genotype of your partner. So that's a dominant disease. And X-linked, I think you all probably know, are carried by the mother, and they are transmitted through the mother. The daughters can be carriers, the sons can be unaffected or affected, and the daughters could also not have the disease as well, not carry the disease. So how do people find out that they're carrying a genetic disorder? Well, they may have other family members affected, so they may realize that through their family, they've realized they've had people affected with beta thalassemia or cystic fibrosis. So as a family, they've got tested, and then they can find out who is a carrier. They may only realize when they've had an affected child, and that's the exact situation we want to avoid. We don't want people to first find out they're at risk when they have an affected child. They may be diagnosed as an adult because they may be ill. So this is especially relevant for dominant disorders. It includes things like Huntington's and various cancers and other late onset disorders. And I'm going to talk to you about preconception carrier testing and preconception carrier screening. So reproductive genetic testing, as I said, we've got the preconception, so before the couple start to have children. I worked for over 25 years on pre-implantation genetic testing, which I'm rarely going to mention today. But so you can test the embryo before the embryo goes back. We can do the biopsy of our embryo in our lab, do the genetic test, and put back embryos that are unaffected. And then we can test a woman when she's pregnant. So, preconception carrier testing. This has been done for decades for groups of people, populations of people who have been at high risk of a particular genetic disease. So it's just normally one disease. Ashkenazi Jews are a little bit more complicated, but for most cases, this is one disease. So, for example, the hemoglobinopathies in Mediterranean and African populations. Now, correct me if I'm wrong, but I did check this recently with some people from Cyprus. Is it still mandated in Cyprus that you have to have the screening? Yes. Yes, right. Everyone else is very quiet. So as far as I'm aware, there's more countries than this where it's mandated. So before you actually get married, they do a test on you specifically, is anyone else nodding, for in this case, beta thalassemia, just so that they're not going to tell you can't get married, but so that they can counsel you on your risk to your future children, so that you're aware and that you're not one of those couples who were unaware until they had an affected child, so you can get appropriate genetic counselling. So expanded carrier screening is really what it says. It's expanding that, but now to look at hundreds of genes. So not just a few, well, not just one gene of a high-risk population. This is looking at hundreds of genes in a low-risk population. So, as I said, we all carry multiple genetic abnormalities. So when you do this, you will find people that obviously will come up in the panel that the company is looking at, their specific genes they're looking at. You will find people that will come up as carrying that disorder. But for preconception testing, it's only important if their partner, as I said, is also carrying the same problem. In the data so far, mainly from the US, they're finding that about 2% of couples are at risk of transmitting a genetic disease to their child, and they were totally unaware before they did the test. And I was at a conference with Karen Sage, who's one of the leading UK genetic counsellors, and she told me that the data she's seen from her experience is about the same, about 2%. You may feel that's very, very low, and that, well, is it really worth testing? But we can discuss that in the panel discussion. De novo mutations, which are things that happen in the embryo, which the parents didn't carry, they just are spontaneous errors that happen in the embryo, which lead to genetic disease. That certainly is a problem, and that cannot be tested for by preconception testing or screening, because the parents don't have that error. It just spontaneously happened in the embryo. So there's a number of companies that offer preconception screening, and I'm going to come back to that when we talk about our study. So we did look at expanded carrier screening a few years ago. My student, Carolina, she looked at many panels. She looked at 16 panels of expanded carrier screening. We never actually published the data about the panels, and I'm going to come back to that. But we did write a paper about what this meant for pre-implantation genetic diagnosis. So in our paper, we said that, as we all know, that if a couple are carrying a genetic disease, their main options, there are other options, their main options are chorionic villus sampling, amniocentesis, and pre-implantation genetic testing. So this is the study we did with Cryos. So Anna Bean and my brilliant student, Molly Payne. Molly did the amazing amount of work of collecting and analysing the data, and Anna Bean and I just looked at it and helped write the paper. But Molly was the real powerhouse on this study. So there were two parts to the study. First, Molly looked at the panel. So she repeated what Carolina had done many years ago, but we'd never published. So she looks at 42 panels that were out in the market from 16 different companies. So some companies had different types of panels. So if we look at the first graph on the left, this looks at the number of unique disorders according to the inheritance pattern. Now, in my view, on these panels, we should only really be looking at autosomal recessive diseases. So these are the ones that are a problem if both parents are carriers. If we start looking at autosomal dominant, so the first one's autosomal recessive, and that was certainly the most common, but autosomal dominant was also done, and also the X-linked disorders, by a number of these panels. If you identify that someone's got an autosomal dominant condition in their mutation in their genes, they are more than likely going to have that disorder when they're older. So you have to be very, very specific about the genetic counselling that you do. And unfortunately, some of these tests you can just buy online and do it yourself and send them the sample. So genetic counselling is not even in place. So I think that's very worrying to do. The next graph is also very worrying. So of the 42 panels, this graph should actually, in an ideal world, be the other way round. So at the very top, it says 42, and it says 0 next to it. This is because there was not one gene, not even cystic fibrosis or beta thalassemia, that was on all 42 panels. How could anyone have an expanded carrier screening panel without cystic fibrosis and beta thalassemia, spinal muscular atrophy, all those common genetic diseases? There were, if you look at number 1, that means that there were over 1,000 panels that had a gene on that was never on another panel, which is crazy. That's totally the wrong way. I always say it's as if the companies had written down all the genetic diseases on pieces of paper, thrown them up in the air, and caught some of them randomly and said, okay, we'll put these on our panel. It's a bit like when we started looking at culture media. I was part of the ESHRE culture media working group, and I again thought all the culture media must have been formed on some scientific basis. No, they were totally different compositions, complexity, etc. And this is exactly the same. So this is not good. What we want is standardization. We want the common genetic diseases to be on all of the panels, and then there might be panels for different ethnic groups depending what issues different cultures have. So this is the Cryos data. So part 2 of the project was to look at the Cryos data, and this was something that Cryos were really interested in doing. This was when Cryos were using their own bespoke panel that for the men, it was called CGT. For the men, it looked at 46 genes, and for the egg donors, it looked at 47 genes. So Anna Bean gave us information on 883 donors. So just looking at 40-odd genes, we found that 17.6% of the donors, 155 donors, had at least one mutation in those genes. And one poor person had 4 mutations. But that's just the luck of the draw. It really doesn't matter. But the regulations for most countries at the moment is that if a donor is carrying a genetic disease, you should not use them. So correct me if I'm wrong, but I think these donors were rejected from going further into being a donor. So you can just imagine now, if we move on to panels that look at 500 genes, that 17.6% is going to go way up. And if we discard all of these donors, we are going to have no donors. And I've been in a conference giving this talk with some people from America, and they said, yes, in our clinic, we rejected almost all the donors. We're like, you can't do that. It's not sustainable to do that. So the arguments against using expanded carrier screening for donors are that these disorders are very rare, it's costly, it's not as much as you think, it varies the cost, and that you reject so many donors, there will be none left. But arguments for using it is that you could prevent a serious genetic disease. But to work, we need to standardize the panels, because if you have a donor that's used panel A, and the clinic is using panel B, and they're not actually looking at the same genes, then that's going to be a disaster. We've got to have standardization. And I think we need to accept donors carrying mutations. We all carry mutations. And we need to genetically match the donors and the recipients. So I'm just going to briefly talk about, finish off by talking about some of the work that we've been doing. As I said, I reported on this paper in 2019 at the Cryos Symposium, but we published a paper in 2016 to say that donor anonymity was a thing of the past because of ancestry testing. And you've already heard from a donor who has identified one of their children from doing that. So, should we tell parents and children of their conception? I think we have to be very open and be honest with our children. I think if children find out by accident, this is a very negative thing. So we have just published a paper on a study we've done with the Donor Conception Network in the UK, looking at these great books that they write to help people find the language to talk to their children. And these, in my view, should be used from when they're a baby, and you put photos of your own family, etc., in the book. So just to finish off, the international fertility education, watch this space. We've got some great news coming very soon. We want to educate the public about fertility, about donor conception, everything, right from school. So we're working on that. And I think you all know that we've got this big gender inequality with the egg and the sperm. But following on from Evelyn's talk, if induced pluripotent stem cells are going to be used to make eggs and sperm, I think Cryos needs to think, how would that be in the future? Would we need egg and sperm donors? So I don't know if it's going to happen in my lifetime. I've always asked Evelyn every time I see her, when do you think this will happen? But this would revolutionize fertility treatment and fertility in generally, especially for women. And this is my last slide. Where do we take the future of humanity? So this is something to discuss at dinner tonight. Have you heard of transhumanists? Well, I have done some work. I was on a TV program with a transhumanist. They don't think that we might need to reproduce at all. We might not need to have children because they want us to use technology, especially genome editing, to edit the human body and our future children to live forever, to maybe become merged with AI, but also to enhance us. So why worry about your poor knee when you just rebuild it or just make someone who's going to have super knees? So is this really the future? Is this where we're going to be? In some of your lifetimes, probably not my lifetime, but it's coming. I mean, I've had laser surgery on my eyes, so am I enhanced? So it's certainly happening. So please reread Brave New World if you haven't read it yet. It's a very short read. The main character's an embryologist. The book was written almost 100 years ago, and it talks about everyone reproducing by fertility treatment. We've got frozen eggs, we've got pre-implantation genetic testing, and I'm sure at some point in the future, artificial wombs. But I think we need public debate, we need legislation, and we need to think about the safety of this technology. I don't know how comfortable people will feel using an artificially made egg or sperm. So in conclusion, I think for genetic testing of donors, we need to match them with the recipients. We need standardized expanded carrier screening. We need to inform the donors and the parents about genetic ancestry testing. And I really hope that soon we will have global fertility education, so people start their fertility journey being fully informed about all aspects, and including donation. I think historically donation is brought into the narrative when they've had failed treatment, and I think it should be really brought into the discussion at the beginning of their treatment, so that it's not just put on the table when they're sort of have failed cycles. And I think some of you follow me on social media anyway, but please feel free. I've done lots of tweets and Instagrams today, and I really like discussing the controversial topics of our field. So would always love to hear other people's views. Thank you very much.